You might recall that DNA is composed of nucleotides. It has two chains of nucleotides held together by hydrogen bonds. The nitrogenous bases are held together in a complementary fashion. That is, adenine is always bonded to thymine, and guanine is always bonded to cytosine. And it's the sequence of nitrogenous bases that holds the genetic code. 3 bases is called a triplet. And this set of 3 bases, this triplet, is the information for a specific amino acid. You've probably heard of a gene. It's the functional unit of heredity, and it's the DNA needed to produce a specific protein. There are 3 types of RNA required to make that protein. One of them is messenger RNA, or mRNA. And all these RNAs are made in the nucleus. DNA is stuck in the nucleus. It can't leave. So it makes small copies called RNA with information to make 1 or 2 or 3 proteins. So the DNA separates. Those hydrogen bonds holding the 2 chains together break, and the DNA strands separate, and a small segment of the DNA, one strand, is copied into messenger RNA. Messenger RNA looks quite like DNA, but it's a single strand, and it contains uracil instead of thymine. Once that RNA is made, and remember that piece of RNA has the information to make a protein or a series of proteins. That RNA will leave the nucleus through a nuclear pore, go to a ribosome to make a protein. On that RNA, the nucleotides basically are in sets of 3. 3 nucleotides as a codon, and a codon is the code for one amino acid in that protein. That process of making RNA, whether it's messenger RNA, transfer RNA, or ribosomal RNA, is called transcription. Once the RNA leaves the nucleus, it will be translated into protein. The production of protein is called translation. There are 3 types of RNA necessary to make proteins. First, the messenger RNA carries the message of the protein, or the exact information of the protein. Ribosomal RNA is used to make the ribosomes, and transfer RNA carries the amino acids to the ribosomes to build the protein. This is a fairly complex diagram. Let's start at the top. First, a piece of messenger RNA, this green strand, is made in the nucleus. That's transcription. And that would be messenger RNA would leave a nuclear pore, associate with a ribosome, and the ribosome will move along that messenger RNA for the process of translation, which is making the protein. Now that ribosome itself is made up of some RNA and proteins, and that would be ribosomal RNA to make the ribosome. Then we have to have a third RNA, transfer RNA. Each transfer RNA carries an individual amino acid. So these little dots here represent amino acids. On the base of this transfer RNA is a 3 nucleotide sequence called an anticodon that will match up with the codon on the messenger RNA. So whatever code is on this messenger RNA is read by the transfer RNAs, and they're going to bring over their individual amino acids, and a protein will be built. Which of the following will appear immediately after transcription has occurred? The messenger RNA. Which of the following will attach to molecules of transfer RNA? Amino acids. With which of the following will DNA nucleotides interact? DNA polymerase. DNA polymerase is the enzyme that helps to build the DNA. We're moving on to cell division. Cell division is called mitosis. It is the process that the cell undergoes to build a new identical cell. So sometimes it's considered to be asexual. So our cells have 46 chromosomes, and every cell, when it divides, must produce 2 identical cells. Meiosis is a different process. Meiosis produces sex cells, which are eggs and sperm, and these only have half the amount of DNA. They only contain 23 chromosomes. The cell itself, when it divides by mitosis, produces 2 new cells. So the cell doesn't die. However, there's kind of a timer in the DNA, and every time a cell divides, a little piece of DNA comes off, and that gives the cell a lifespan. That is, the cell will only divide so many times. And this programmed self-destruction is called apoptosis. The cell cycle is the life cycle of the cell. Just as we're born, we age and grow, and then we die. The cell is produced through mitosis. It increases in size, and then it divides to form 2 new, smaller cells. Well, most of the life of the cell is spent in interphase. That is, it's not dividing. It's just doing what the cell normally does. If it's a stomach cell, it will be producing hydrochloric acid. If it's a pancreatic cell, it will be producing insulin. So the cell is just doing what it normally does for most of its life. And then mitosis will occur, which is pretty quick, and it will be divided into 2 identical daughter cells. So mitosis itself is division of the nucleus. That is, the chromosomes are distributed to the daughter cells. Cytokinesis is the division of the cytoplasm. So there's a physical separation of the 2 daughter cells. So you can see in the blue that most of the life cycle of the cell is in interphase. It is not a part of mitosis. And then the cell undergoes mitosis, or division of the nucleus, and then cytokinesis, division of the cytoplasm. Well, before the cell divides, the DNA has to replicate. Remember, one cell is going to become 2 cells that are identical, so it must double the amount of DNA, and this occurs during interphase. The enzymes unwind the DNA strands. Remember, those 2 DNA strands are held together by weak hydrogen bonds, and DNA polymerase is the enzyme that is going to bind to the exposed nitrogenous bases, and it's going to allow the bonding of the complementary base to come in and bind to the DNA strand. So here you can see DNA. Part of it on the right hand side has been unwound, and that's once it's unwound, then these complementary bases are exposed, and new bases will come in to make an exact copy, using the old strand as a template. At the end of DNA replication, the original strands have completely separated, and there are 2 new identical DNA molecules. Not only that, but proteins have been synthesized that are necessary for replication. The centrioles have copied, and they're ready for replication. And the cell will leave interphase and go through mitosis. So here are 2 identical DNA molecules after replication. There are 4 phases of mitosis. Think of PMAT, P-M-A-T. In prophase, the replicated centrioles move to opposite sides of the nucleus. It is these centrioles that the spindle fibers will basically grow from to produce the spindle apparatus. In prophase, the nuclear envelope disintegrates, and the chromatin, which is the stretched out form of DNA, winds up or coils very tightly to become a chromosome. The second phase, metaphase, think of middle. The chromosomes are in the middle of the cell. Anaphase, A for apart. The chromosomes pull apart from each other to go to opposite sides of the cell. And telophase is basically the opposite of prophase. The nuclear envelope reforms, the chromosomes uncoil to become chromatin, the spindle apparatus is going to disappear, and the cytoplasm constricts, forming the cleavage furrow. So in animal cells, there's a cleavage furrow. It looks different in plants, and that cleavage furrow is over here. See telophase. You can see this, how the cells are starting to squeeze apart here. That is a band of proteins that's kind of squeezing them, and that is the cleavage furrow. So the stages of the cell cycle are interphase, and that's the normal life cycle of the cell, and then the cell goes through prophase, metaphase, anaphase, telophase, and then cytokinesis. These are not a part of interphase. Cancer is a problem with this whole process. That is, mitosis continues abnormally. A tumor is a mass produced by abnormal cell growth. Benign tumor is a mass that stays in place and is not cancerous. A malignant tumor is a tumor that's dividing rapidly, but it's changing. It stimulates blood vessel growth to feed it, and the cells can break off and go to other areas of the body, and that is called metastasis. Malignant means, mal means bad in French and Spanish. The malignant tumor is the bad one. And this shows the process. So cancer begins with one cell that's dividing abnormally. And here's that one cell. Here are the offspring of that one cell. And when it stays in place, it is a benign tumor. But a malignant tumor will break off and spread through the bloodstream or lymphatic system. And it will also cause blood vessels to grow to the area to feed the tumor. Malignant cells are not normal. They don't function normally. So a malignant lung cell would not allow in the diffusion of gases. A malignant tumor of the thyroid gland produces abnormal amounts of thyroid hormone. And here is a thyroid tumor. Which of the following is not associated with interphase? Prophase. It's part of mitosis. Which of the following is a passive mechanism for moving solutes across the cell's plasma membrane? Well, we haven't gotten to this yet, so this question's a little early. But we'll tell you right now it's diffusion. Which of the following does not move through the phospholipid bilayer of a cell's plasma membrane? Again, we haven't gotten to this one yet in the PowerPoint, but that's okay. Glucose will not move through the membrane. Just about the only thing that moves through the membrane is gases and small lipids.